So next invited speaker is Professor Paul Weiss. He is a nanoscientist and holds a UC presidential chair and is a distinguished professor of chemistry, bioengineering, and material science at UCLA. He studies the ultimate limits of miniaturization, developing new tools and methods for timing resolution and spectroscopic imaging and chemical patterning. He applies these advances in other areas, including neuroscience, microbiome studies, quantum information, and high-throughput cellular therapies. He has won awards in science, engineering, teaching, publishing, and communications, and he's a fellow of the AAAS, ACS, AA, MB, APS, AVS, Canadian Academy of Engineering, Chemical Research Society of India, IEEE, and also MRS. And he's the founding and the current editor-in-chief of ACS Nano. Please welcome Professor Paul Weiss and his talk.
Well, thank you so much for having me and for that wonderful introduction. I'm sorry not to be able to be there in person, but I will look forward to visiting next year. As some of you know, I have a joint center with SKKU and NTU in Singapore, so many affiliations there. What I thought I would do today is talk about an aspirational part of our laboratory, in which what we'd like to do is have optimized, atomically precise contacts, with the idea being that those contacts are simultaneously chemical, physical, mechanical, and electronic, and if we play our cards right, also have spin control, and that we can design these from the bottom up, hopefully develop a predictive understanding of them so as to develop precursors that give us the same interfaces every time. And let me start with an almost trivial example of graphene, where right now what we do to make contacts to this 2D material with its extraordinary properties in its semi-infinite form is we evaporate metal on top of it, which destroys those properties, or we oxidize it, breaking the conjugation, and then reduce it back a little bit, but still, again, at the contacts, we've destroyed the extraordinary properties. And so what we argue is that we should be able to use more than a century of organic chemistry to make an atomically precise contact in defined and predictable ways to optimize the transport across an interface. We have the scanning probe microscopes and the spectroscopic capabilities you saw so beautifully illustrated in Mark Herson's talk in order to map the molecular orbitals across those interfaces. Now, there are other interfaces that are buried that'll be more difficult, but in fact, we have developed the tools in order to study those. And in this work, we've pulled in a host of collaborators, and I'll talk about some of their work here. If you're interested in some of the latest work along these lines, I put these papers up here. Okay, now to back up a little bit, and part of my role in ACS Nano and in the nanoscience community, we were asked at the 10th anniversary of the US National Nanotechnology Initiative to say what it is we had done with more than $1 billion per year in funding, and what we would do if we were given another 10 years of funding. So those seemed like two very important questions to answer, and we took it very seriously. We went and looked and realized that one thing we're able to do is make atomically precise, quote unquote, perfect structures, study their properties, vary those structures, and understand those variations. And that's really the subject of today's talk. We also realized that function at the nanoscale, even in these atomically precise structures, and especially in biology, is heterogeneous, and that turns out to be very important. And in terms of those two, scientific results, as I said, we'll talk about the first one today. The other one's a different talk, and we can cover that some other time. We also realized that the main advances in the biotechnology revolution came from the development of new tools that allowed us to ask new questions, and that we should be doing the same thing in nano. And then perhaps the most surprising result was that we realized that unlike other fields, we had developed communication skills because of the way nanoscience had developed. That people from chemistry, physics, engineering, math, medicine, toxicology, and so forth all came together, and we shared problems, and we shared approaches, and we developed new approaches. And for whatever reason, other fields had not done that. And so for the students and postdocs here, this is the Young Scientist Symposium. I want to tell you that you're getting training that people in other fields, your colleagues elsewhere, don't have, and you can use that to advantage in what you go out and do in the world. And it's the reason that the BRAIN Initiative in the United States came from our community and a committee that I'd put together for the White House. And the Microbiome Initiative, likewise, was put in the hands of the nanoscientists because we were inclusive in asking other people to work on these problems. So we published a series of papers about that, as well as this one on how to take what we learn in clinical medicine and develop engineering and nanotechnology to address those problems. Okay, now once upon a time, back when I was at IBM with Don Eigler, I was the first one to move atoms around. And really there, I just wanted to find out what was underneath them. Why were xenon atoms sitting out in the middle of a platinum 1-in-1 terrace? But that really led to the idea that we could position atoms, we could make precise structures, we could study their properties. We didn't do graffiti at the time or anything, or later, after I left, Don Eigler built this quantum corral, which has the interesting property that from electron interferences, we can see protrusions or enhancements of the molecular orbitals at particular energies and positions, and that actually affects the chemistry. And so what we realized from that, and from some earlier very beautiful work by Joe Strosio and Randy Finstra at IBM Yorktown Heights, when they looked at the stoichiometric gallium arsenide 110 surface, where there's charge transfer from gallium to the more electronegative arsenic atoms, that the filled states ended up on arsenic, the empty states on the gallium, and we could effectively put on goggles and look at the surface the way a mobile atom or molecule would see it. And so in terms of understanding chemistry, and here in terms of understanding electronic properties, and what you saw in Mark Herson's talk as well, that gave us this opportunity to explore the surface energies and the surface electronics and ultimately other properties like spin by developing the tools to measure those orbitals and contacts and so forth with atomic resolution. Okay, so I gave you the example of graphene where the interfaces are exposed. Another example is in transitional metal chalcogenides. So Taki Lee at Seoul National University passivated vacancies in molybdenum disulfide by filling the sulfur vacancies with alkane thiol molecules. We'd like to take this a step further, where instead of just having a passivating structure, we fill in such a vacancy with a molecule that then serves as a contact. And in fact, I don't have time to talk about it today, but there are boron-containing molecules that are aromatic, where we can control the charge on them that we think will serve this purpose. And if you're interested, please ask. We've put together a series of papers on that and are working actively in that area now. Now, the way we got into this was we started looking at a number of different systems as a way to probe contacts. In the very early days of this field, we looked at these self-assembled monolayers of organic molecules terminated in sulfur on gold surfaces. That'll be familiar to many of you as self-assembled monolayers, first developed by my Bell Labs colleagues, Dave Valera and Ralph Nuzzo. Dave moved a little bit ahead of me to Penn State. Ralph moved to the University of Illinois. And at the time, it was thought that these molecules that tilted over and formed these one molecule thick two-dimensional crystals went down randomly and irreversibly and were immobile on the surface once they got there. Well, we had developed a spectroscopic imaging tool that we thought could differentiate between different molecules. But even before we turned on that capability, conventional STM showed us that those three ideas about the irreversibility and the immobility and random adsorption were incorrect, that the molecules phase separated, the monolayers were mobile, they never reached equilibrium, that there was domain coalescence to reduce the total interface length, which was energetically unfavorable. And over the years, we developed the ability to image every individual molecule, to image the domain boundaries, and then by directed assembly, control the type and density of defects on the surface, and then to insert molecules that we wanted to study either individually, in pairs, in lines, or in two-dimensional clusters into those interfaces. And then we could position the probe tip of our microscope to study their function, spectra, and structure. And so one of the first systems we studied were these molecules that were known to be conductance switches in bundles of 10,000 when they were inserted into pores in silica nitride membranes by the late Mark Reed at Yale. And actually, Taki Lee was his postdoc at the time. And so by isolating individual molecules, we showed that individual molecules could function as switches. And then through more detailed experiments, we figured out that the mechanism was where the molecule of switching was where the molecule tilted. And thus, it was that buried contact that was everything in terms of the switching. So just like semiconductors, the contact was what really mattered here. And so we asked three questions experimentally. How quickly could we change from one state to another? What's called the slew rate? What's the conductance of the buried contact? And what are the absolute structures? And in each case, we developed tools that could answer those questions that we're going to apply to further experiments in contacts. So in terms of the absolute structures, what we wanted to understand was where, what was the configuration of the molecule. And by looking at a series of mixed monolayers of longer and shorter chains that formed identical structures, we could show that the topographic measurement in the STM, if we kept the tip out of contact with the surface, measured the outermost layer, but we could simultaneously measure the buried contact by virtue of its dipole, and in that way get the absolute azimuthal and polar tilt angles. It was actually an experiment that took us 20 years to make work that we tried by a variety of methods before we eventually hit gold, so to speak. Okay, we can also look at buried interfaces. That's really part of a different story, but we have capabilities of looking at several different buried layers and using that to determine with atomic resolution the structures of biomolecular assemblies such as amyloid-forming peptides, as one finds in, for instance, in Alzheimer's. Another thing we could do is we could measure the polarizability of the molecules. And in this case, while we get molecular resolution, we include in that measured polarizability these substrate electrons that can go in and out of the molecule at the frequencies which we measure, which are in the microwave region, so up in the gigahertz. And in this case, we can look at the same switch molecules in the on versus the off state, and thus measure how open that conductance valve is that serves as the switch. And in that way, we have a way to measure buried contacts. Now let's move over to graphene for a few slides. And this is Patrick Hahn, who was the student who previously measured the tilt angles. He had an interest in chiral graphene nanoribbons. Those hadn't previously been studied. A number of groups had studied the armchair and the zigzag nanoribbons, and those were known, for instance, for the armchairs, that when you reduce the width from the semi-infinite planes to these finite widths, that the narrower one went, the larger the band gap one got. And if one wants to have semiconductor properties, then a band gap is necessary. And for the zigzag nanoribbons, the edges have interesting spin properties, and Patrick thought we should be able to find interesting spin properties as well for these chiral graphene nanoribbons. So in fact, there's a chemistry that's been known for over 100 years called Ullmann coupling, in which aromatic molecules are put together by starting with haloaromatics. It works with 100% efficiency in vacuum or solution on copper surfaces. And we had previously in our group figured out how these molecules interacted as reactants on the surface, how the reaction proceeded through intermediates, and thus Patrick was able to predict which precursor to use to end up with the same chiral graphene nanoribbon every time. And really the leader of this field is Klaus Mühlen at the Max Planck Institute. And he'll come in to play a little bit later. Okay, and the reason we use the copper surface is that it interacts strongly with aromatics. The molecules sit close to the surface. They're distorted by their interaction with the surface. Compare that to gold, where aromatics sit far off the surface. And in earlier measurements, we looked at benzene on the gold 101 surface. We could show with STM that we could peer right through the benzene molecules in some cases, and the gold 101 surface wasn't reconstructed, meaning the interaction was rather weak. Silver, there's an intermediate, but still relatively weak interaction. But copper, there's a strong interaction. It breaks the carbon halogen bond, and it satisfies the bonding that would otherwise leave a radical on the surface from where the carbon atom lacks its fourth, its full bonding. And so what Patrick did is he showed that we could measure the precursors, which formed complexes on the surface. He could raise the temperature to where the halogen was cleaved off, and then go back to low temperature and measure that, and then go up again to where the intermediates coupled together, very much like the arrow pushing that we see when we take or teach organic chemistry, and that we're able to get these chiral graphene nanoribbons. And so he showed that according to the growth direction, one got alternating left and right, oops, that arrow moved over somehow, left and right chirality of the enantiomeric graphene nanoribbons. A former postdoc in the group, Nacho Pascal, took a vicinal surface in which the terrace width was narrow enough that everywhere on the surface, he only got a single chirality, and some really beautiful experiments that he did in San Sebastian. And as I said, we can do these spectroscopy along these graphene nanoribbons. We can do things like push a bromine atom up to the edge of the graphene nanoribbon in order to turn what's basically a particle in a box into particles in two boxes by quenching the electronic state within the molecule. Now, when we published this work, someone wrote a comment about it saying that under different conditions, at higher temperature and higher coverage, that they got different products, and suggested that our mechanism might be incorrect. And so we took that comment seriously. We reproduced their conditions, showed that indeed we got different products, but we had high resolution STM images than they did, and we showed that our mechanism was still correct. But what got me excited here was that when I looked at these joints, I realized that those were contacts, and that we could probe those spectroscopically with atomic resolution, and we could look for what would make a good contact. And so by taking their comment to heart, we were able to jump our research forward, but by what I estimate was about 10 years. So we had this idea in our head that we could do it. We had no idea how to proceed. And then, what we see in the future here is designing a combinatorial set of precursors that would give us many different contacts on the surface. We could measure all of those, couple the theory, test, and improve the theory to the point where it could be predictive. And then we could design precursors that would snap together to give us the precise contacts we want every time. Now, we haven't done all that. What we have done is start looking at the contacts that were formed on the surface. And you can see for the straight graphene nanoribbon, we do quite well, right? There's a continuous wave function. When we look at what's called differential conductance, there are no nodes, right? If you had a node and a contact, that would be very bad. And if we just look at the topographic images of, for instance, this graphene nanoribbon, it looks okay. But when we measure the differential conductance, we see there's a node right in the middle. So that would be a bad contact. And, for instance, in this one, right, all these stripes are the kind of things that give me nightmares that would make a terrible contact. And so we're still on the road to figuring out how to make these contacts in an optimized way. We now know how to measure them and know how to, what the figure of merit of a good contact is. I'll point out that Klaus Müller and Roman Feisel jumped in this controversy as well, and they showed with non-contact AFM measurements that indeed, we got the structures we said we did and that we had the mechanism correct. It was nice of them. Okay, now one of the things we can do is we can functionalize the sides of the graphene nanoribbons effectively to dope them. We've done that with sulfur, and other groups have done that with nitrogen. My colleagues here at UCLA, Rick Kainer, Anastasia Alexandrova, Ken Hauk, Sarah Tolbert, and Eve Rubin did that as shown here. And then another one of our editors, Andrew Wee at National University of Singapore with Kianping Lo, who's on our advisory board, also did that and showed the assembled structures on the surface through their electronic interactions with this nitrogen doping. So there are a number of tricks that we can play in order to manipulate the electronic structure. There are other parts of the talk, if I had a little more time, where I could describe the side group functionalization, the control of charge within CADE structures and molecules, where I could illustrate that. The Berkeley group has done some really beautiful work here in making armchair graphene nanoribbons of varying widths in line so as to make, quote unquote, 1D device. Another thing they did is side group functionalization to manipulate the electronic structure so that they got a device with this, chemical physical borderline, and they are able to shift the bands according to the side group functionalization in these two different zigzag segments. And let me just tell you in the last couple of slides where we're headed in terms of spin. So it turns out that chiral molecules function as spin selective transport in a process called chirality-induced spin selectivity, discovered by Ron Nauman at the Weizmann Institute. He showed that in photoelectron spectroscopy and DNA. Many groups have since looked at this in a number of different biomolecules and synthetic molecules. There's the thought that this may play important roles in spin in biology and what's called quantum biology. And one of the leaders in that field is a young colleague, Clarissa Ayo, here at UCLA in electrical engineering. And the idea of having well-defined chiral systems that are robust may enable us to take spintronics up to, or at least close to, room temperature instead of the ultra low temperatures that are used now. A lot of these ideas are in a paper that we hope is going to appear in ACS Nano soon. I'm working on the minor revisions, but it's already up on archive if you'd like to read it. Okay, and in our group, what we've done is measurements in a number of different materials and scales. So for instance, pattern DNA, where we can do 100,000 measurements simultaneously in looking at pattern DNA and pattern fluorescence, where we have magnetically controlled substrates, and then spin polarized low energy electron microscopy and electrochemistry, and now scanning probes. And we've looked at DNA, we've looked at chiral peptides, we've shown in hairpins we can reverse the chirality in addition to reversing the magnetic field, and we're figuring out how to quantify these effects, led first by John Abendroth, who's now at ATH, and Dominic Steamer, who's now at Fritz Haber Institute. Okay, so I try to give you a glimpse of how we're thinking. We don't have the punchlines yet, but there's really room here for people from chemistry, physics, materials, engineering, experiment theory, simulation, to move this field forward. It's a rich area with very significant consequences if we can make it work, right? If we can lower the resistance of contacts, then we can make more powerful, laptops, phones, and so forth that don't melt our legs off when we carry them around. And they're just, I think, tremendous opportunities for this field. And I look forward to, more discussions when I'm able to come and visit there. I tried to put everyone's name on every slide as we went along. Here are the people who paid for this work, especially NSF, DOE, Fulbright, and Keck foundations. Collaborators around the world, including SKKU, where I'll be hoping to visit soon. And, really the idea here is that we have this opportunity of control. We can leverage, these many decades of chemistry and the developments in material science and our ability to visualize these contacts, whether they're exposed or buried. And so I think I'll stop there and try and leave a little bit of time for questions. Thank you.
Hey, thank you very much, Paul, for your wonderful talk. And then let's take a few questions from the audience. Any questions?
Hi, Paul.
Hello,
Lisa. Thank you for your wonderful talk with the beautiful images. It's always nice to see your work. So I was wondering if you have had a chance to integrate some kind of like machine learning techniques to process your atomic level images. So these days, so many, so numerous imaging techniques based on machine learning, so many things. Every day something new pops up, but it doesn't sound really easy to integrate those techniques at the atomic level, especially for the imaging. So I was wondering what's your idea on this?
Yes, we work very closely with mathematicians here. Two of the leading mathematicians in the world are Andrea Bertozzi and Stan Osher. So we have Terry Tao in our institute, who was the one who showed that the Nyquist theorem was wrong, and that as long as your data are sparse, right, then you can dramatically reduce the amount of data you record to get all the information. It's not like compression, right? And so, for instance, we used that collaboration in determining the structures of the amyloid-forming peptides, where we fused the different modalities together. The first time we recorded a structure, it took us 2 years, and we didn't learn anything. But just using the analysis side of that collaboration, we've been able to reduce it to 10 minutes. So it's a 5-order magnitude increase in efficiency. Now, the next step is to do guided acquisition. So you only go and record contextually where you can most efficiently constrain the geometry, for instance, in that case. And so it's a very valuable tool we have. And I tease my group that no one wants to record STM images unless there are 3 mathematicians sitting next to them. Oh. Now, it's not quite that bad. We've put all those algorithms up online and anybody can use them. And actually, I'm just working on a review paper that's due in the next 13 days. We'll send to JPhys Chem. But there are a number of papers that include Andrea Bertozzi, Stan Osher, and then one of their progeny, Jerome Gillis, who's now at San Diego State, where we work very closely together. So I briefly flashed that the amide containing monolayer, there, the way that we got the positions of both the sulfur gold bond and the hydrogen bond from the amide was from that mathematical analysis. And actually, there's a big surprise there, and that was that the hydrogen bonding network that's buried continues not only across domain boundaries, but also across regions of disorder. So it explains some data we had where we'd put those amide-containing files on liquid metal nanoparticles, and then when we excited them with ultrasound, we were able to make smaller and smaller particles without oxidation. Whereas if we had the monolayer without the amide, they just oxidize into, basically gallium oxide and indium oxide. Yeah. Okay. So it's a very, very valuable, and something we really want to push further. We actually just submitted another proposal to keep working with them.
Okay, we'll look forward to your next talk next year then in
Korea.
Thanks
very much. Yeah.
Okay, do we have any more questions? Okay, then thanks again, Paul, for your
My
nice talk.
pleasure.
Okay, then let's move on to the next speaker, Dr. 서경 Yoon at 성균관 University. Today, Dr. Yoon will talk about surface polarity insensitive organosilicon-based clustering of nanoparticles with intragap distance tunability. Okay, once you're ready, let's start your presentation.
So, hello everyone. I'm Seogyung Yoon from Sungkyunkwan University, and it's a great honor to give my talk here in conference in the Korean Vacuum Society. Today, what I'm going to talking about is synthesis of surface polarity insensitive organosilicon-based clustering method. So to give you a brief background of my research, I give an explanation about the colloidal nanoclusters first. So thanks to the incredible advances in the nanofabrication technology, we have a variety of nanocolloids, and based on those nanoparticles, a lot of efforts have been dedicated to develop a composite nanomaterials, what we call here the nanoclusters, made out of different functional nanoparticles. And presently, colloidal nanoclusters have 3 different types according to their morphology. Colloidal superlattice is usually the closely packed structure like this, but non-superlattice nanocluster is loosely packed structure like this. Because the colloidal nanoclusters stem from the studies of superlattices, so majority of studies have been done when it comes to the colloidal nanosuperlattices. So what we focused on was to develop a method to synthesize a non-superlattice nanoclusters to avoid unexpected interactions and to enhance the gap distance tunability of embedded nanoparticles. So this scheme is showing about the clustering method. So in this work, we developed a new silica-based material. Here we call it organosilicon, OSS in short. So in order to synthesize the OSS, what we need are only the surfactant solution, here the CTAP, and the THMP, which is a kind of basic monomer of organosilicon. So when the THMP is injected into the CTAP solution, then the Tyndall effect is observed, indicating OSS is synthesized. This scheme is describing the entangled molecular structure of OSS. These TM images are showing that the shape of OSS is nanoscaled sphere. Due to the polymeric property of the OSS, the structure was collapsed and agglomerated very easily under vacuum and the dried condition for TM visualization. By contrast, cryogenic TM images is showing intact spherical structure of OSS. And owing to the CTAP on the surface of the OSS, it has positively charged surface, and the DLS data confirms the size and the monodispersity of the OSS again. Next, we identified that the gold nanorows here as a model system, in short GNL, could be co-encapsulated in the OSS and assembled into the spherical cluster shape successfully when the OSS synthesis process was done in the presence of GNLs. This is a cryogenic, and this is a just common GM image of GNL clusters. So GNL nanoclustering process was monitored by the UV visible spectroscopy because the longitudinal plasmonic resonance peak of GNL is very sensitive and highly effective by the distance between the GNLs. So after the injection of the THMP into the GNL solution, a curve shift was observed, implying the clustering of GNLs. And we found the clustering manner was different according to the concentration of CTAP here. And basically, siloxane bonding between the THMP is reversible, and the rate of reverse hydrolysis is elevated in high pH condition. So we hypothesized that the GNL clusters will be condensed under high pH, resulting like closely packed structures. So we confirmed that the pure OSS could be dissolved out after adding the sodium hydroxide solution, but the OSS without the sodium hydroxide solution maintained its structures. So we monitor the clustering, and this solution stays like this. As you see here, the color of the GNL nanoclusters turned into like dark brown after the injection of sodium hydroxide solution. And cryo-TEM images reveal that the density of the GNL nanocluster is a little bit higher in stage 2 than those of stage 1, indicating that the dissolution of the OSS layer was a result in the closely packed structures. Next, we solidified resultant GNL nanoclusters using silica to freeze its spatial distribution for further applications. To do so, we injected TIAS as a silica precursor with 1 hour interval in between. As a result, gap distance between the embedded GNL was controlled according to the injection sequence of THMP here, which is a clustering inducer, and the TIAS from closely packed one to the loosely packed one. And we found another interesting thing, in case of GNL silicon nanocluster, the orientation of incorporated GNLs are controlled by the size of nanocluster. So when the size of nanocluster was smaller, less than like 70 nanometer, it shows a parallel order, but it shows a randomly ordered structure at the counterpart size. To prove that this is not a coincidence of few examples, we measured DLS, and it shows a diminishing rotational diffusion peak here as the size of GNL clusters increased, indicating that the orientation is randomized as the size increased. So based on the results, we carefully controlled the size of nanoclusters, and we successfully synthesized the orientation controlled GNL nanoclusters. And this is important because the plasmonic coupling mode is different according to the assembly structure of GNLs. And this chart is showing that the number of nanoclusters that contains more than 5 GNLs inside of it, and it turned out to be that it was possible that the particle loading number can be regulated by the size of nanoclusters. So, and we identified that this clustering method worked regardless of the shape. So we demonstrated spherical GMP, gold nanoparticle nanoclusters. We controlled the spatial distribution. And finally, we focused on the fact that the nanoparticle obtained from the non-polar solvent can be transferred into the water by using the CTAP as the second surfactant. So what that means is that the nanoparticle with different surface chemistry can afford OSS-based clustering methods, since it's difficult to put 2 different nanoparticles like aqueous-based and organic solvent-based one into one nanocluster together due to the difference of the surface chemistry. So we used water transferred superparamagnetic ion oxide nanoparticle and the quantum dots, which are very most representative functional nanoparticles obtained from the organic solvent. So their inherent superparamagnetic property and the crystalline structures of ion oxide nanoparticle and the optical property of quantum dots were still sustained after water transfer. And we have succeeded to synthesize a quantum dots and ion oxide nanoparticle clusters respectively. And based on the gap distance tunability and the universality of the clustering method, we synthesize a nanoclusters with multiple functionalities by introducing the GNLs and the ion oxide nanoparticles and the quantum dots at the same time. And this is important because it was difficult to synthesize a cluster with both fluorescent property from quantum dots and the plasmonic property from the gold nanoparticles due to the optical interference between them. So we were able to synthesize a fluorescent nanocluster by tuning the gap distance between the quantum dots and the gold nanorows far apart like this. And we can see the fluorescent intensity of the nanoclusters can be easily controlled by tuning the distance of embedded nanoparticles like this. So this nanocluster has fluorescent imaging modality like this and the photothermal effect at the same time. And furthermore, the MR imaging was able to be conducted due to the presence of superparamagnetic ionoxide nanoparticles. So in summary, owing to this OSS, we created a new type of non-superlattice nanoclusters with high tunability and the universality over embedded nanoparticles. Thank you for listening to my presentation. So I will stop here, I think. Yeah.
Okay, thank you for your nice talk. And then, do we have any questions from the audience?
I have one question. Can I ask a question?
Okay.
Yeah. Can you explain a little bit more detail about OSS?
Okay, about the formation mechanism, or which component I've used, which one?
Actually, I could not perfectly understand, so a little bit. And we don't have enough time, so just a little detail.
Okay. So basically, the OSS is kind of the spherical nanocolloids, which is suspensible in the water, suspended in the water. And its components is only two things, which is a THMP and the CTAP, which is a surfactant, specifically the cation surfactants here. So the method to synthesize the OSS is quite easy. So as I told you before, like first we prepare the CTAP solution with specific concentration, and just add some THMP with specific concentration again, and just stir it. Then we would get like this kind of solution that contains the OSS inside of it. And their molecular structures is like this. Actually, the THMP can form some chain-like structures by making some bonding between them. And the CTAPs, which is the surfactant, can be intercalated in between this THMP chains. So if we're making like this kind of structures, we will get this kind of the OSS collider solution.
Okay, thank you very much.
Okay, please go ahead, Paul.
Yes, that was lovely talk. I was curious how far you could take the pre-organization of the nanoparticles. Could you program in something that you could then keep all the way through to the encapsulation? You showed, some aspects of alignment that could be achieved, but I'm just wondering if you've checked how far you could push that.
Okay. Well, actually, I haven't tried how far I can control the gap distance between the nanoparticles yet, because that will intuitively that would depend on the the final size of nanoclusters, right? So in this paper, we've just like tried to synthesize, we just try to increase the size of nanoclusters up to like 200 nanometers. And usually the most of the size of the building block nanoparticle that I've used in this paper was like 10 nanometer to like 30 nanometers. So theoretically, by using this method, well, I can synthesize like hundreds far away control the nanoclusters, I think. So that's going to be my answer for now.
Thank you.
Okay, so I have a question, please.
Okay.
Okay, so thank you very much for the very nice talk. So you just mentioned about the very nice synthesis of nanoparticles, but how do you think it will be possibly be used? So whether it can be used as kind of a source template, for example, because nanoparticles are concentrated in the middle. So we think it might be possible to kind of enhance the optical signal, for example. So it would be very nice if you can kind of mention about it.
Yeah, thank you for the good question. Actually, that is the thing exactly what we are working on it for the further work of this project. Well, actually, as I've shown you before, like, so we have used a gold-based nanomaterial usually, which is very effective when it comes to the optical applications. So now we are trying to design some source kind of antenna material by introducing like 2 or 3 or 4 like controlled number of gold nanoparticle inside of it with some controlled distance between the gold nanoparticle, because in order to utilize there's a source phenomena, which is very highly affected by the distance between nanoparticle.
Okay, so due to the time limitation, we have to move on to the next speaker.
Okay, our next speaker is Professor Yang from UCLA. Professor Yang, are you there?
Yes, I am here.
Thank you for coming today. So
Yes,
would you please share your slide?
Yes, I do that.
In the meantime, let me briefly introduce Professor Yang. Professor Yang received his BS in physics